Hey everyone, hope you're doing well today. Here we begin the last full chapter of Unit 6 looking at biotechnology. Now, in terms of what biotech is the use of different sort of technologies or tools to sort of be able to manipulate what cells produce and give us the capacity to sort of study what kind of function cells can perform. A cool example of this is the study of bioluminescence that was pioneered by the gentleman we see here, Emmett Chappell. He did all sorts of work for NASA and the Jet Propulsion Lab back in the 50s, 60s, 70s. And as part of the Viking mission to study Mars, he did work with bioluminescence. What he was able to figure out is that if you combine these two proteins that you find like in a firefly, like luciferin and luciferase, if you combine those in the presence of ATP, it will produce light. And since all life uses ATP, it becomes a quick test for life. So all sorts of medical tests will use this. We have the mission to Mars. We're going to bring back samples of Martian soil within the next decade. You'll be able to use techniques that Dr. Chappell developed in order to look at seeing if there's life in Martian soil or there's evidence of that. Again, another cool thing about this is that you're able to use this capacity for bioluminescence to test for the presence of um, cancer markers in cells. So anytime you look at images and you see like the little glowing images, that's examples of biotech that were developed by Emmett Chappell originally. All right, so we're going to look at all sorts of different types of biotechnology. The first example we're going to look at is genetic engineering that occurs using prokaryotes as the tool or as the vehicle for this. Recombinant DNA technology combines DNA sources from two different organisms or two different species. Because DNA is a universal code, what you can do is sort of basically use cellular machinery interchangeably between different organisms. So you're able to take like a gene of interest and put it in some bacterial DNA and then get the bacteria to express that gene or create some product. Where the genes and bacteria are typically inserted are into little rings of DNA called plasmids. Okay. So when we eventually get into our glowing bacteria lab, what you'll do is you'll take a gene from it's from a squid. You'll take a gene from a glowing squid species, and then you will insert that into the E. coli, and you get the E. coli to produce the glowing protein, and then they'll biofluoresce, they'll glow. Here we have a quick animation over recombinant DNA and how that's created. So let's watch that.
A common technique in genetic engineering is to insert a new gene into a loop of bacterial DNA called a plasmid. The molecular tool used to cut DNA is a restriction enzyme such as EcoR1. The enzyme has a precise shape that allows it to run along the groove of the double helix, scanning, in the case of EcoR1, for the base letter sequence GAATTC. The enzyme then cuts the plasmid at this specific point, allowing a new piece of DNA to be inserted. When it cuts, EcoR1 leaves a sticky end.
Notice the overhangs.
This helps the new gene to attach. The joins are then stitched together by another enzyme called DNA ligase. The genetically engineered bacteria is grown in a culture medium. Very quickly, large numbers of the bacteria can be produced, each with a copy of the inserted gene. The bacteria duly manufacture whatever protein the gene codes for, and so the desired product is made.
All right, very cool. So it's a great way to get bacteria to produce some sort of molecule of interest. I don't know if you've ever seen stuff where people talk like about how like certain types of plastic utensils are made from corn or something like that. Or sometimes like the disposable lunch trays that people may get, the sort of foamy looking lunch trays. They'll say it's made of corn or made from corn. What they do is they feed the corn to bacteria, and then bacteria crank out the polymers. Or my friend Chris, his work right out of college was making recyclable plastics. Well, E. coli made the polymers. E. coli made these plastics. So what you do is you take genes from different species and you get those into the bacteria, and then the bacteria can produce the molecules that you want. All right, so let's look a little bit at how this works. They mentioned some of this in the animation. So there's a certain type of enzyme. It's called a restriction enzyme. They're also called restriction endonucleases. Either way, it's talking about the same thing. And again, in the animation, they mentioned enzymes, they're really particular. So in the case of EcoR1, it looks for a specific sequence of DNA and then it cuts it. Now, what's really cool about how restriction enzymes cut DNA is that they make staggered cuts like this. So, what these staggered cuts do is create these overhangs that are called sticky ends. And the important thing about those sticky ends is you've got these single bases that are exposed, and then they can hydrogen bond with complementary sticky ends from a different molecule. So restriction enzymes are really great for a couple of reasons. One is they cut DNA at very specific sequences. And the other benefit is what they do is they cut DNA in such a way that it has these staggered cuts that create these sticky ends and allow you to join DNA from two different sources. Now, why do bacteria have these enzymes? That's a great question. Remember, the oldest enemy for bacteria are viruses, and viruses have the capacity to insert their viral DNA into bacterial cells and kill them. So, what bacteria have evolved to fight against viruses inserting their genetic information is to have these restriction enzymes or these restriction endonucleases. So, if a viral uh, capsid or if a virus tries to infect a bacterial cell, if the viral DNA has sequences that are recognized by these restriction enzymes, then what the bacterial cell does is it basically cuts up the viral genetic information before the virus can take over the bacterial machinery. So this is again, it's a defense mechanism for bacterial cells. They have these particular enzymes that can spot certain sequences of DNA and cut them up. In that way, they can cut up DNA that doesn't belong to the bacterial cell. So let's look at how we can use this. Okay. Now, what is really helpful, again, is the fact that the genetic code DNA is universal. All forms of life on Earth are using this A, T, C, and G code. So because of that, you can use the same restriction enzyme to cut DNA from two different species. And when you do that, they'll create, again, those complementary sticky ends. So then what you can do is join together or anneal DNA from two different organisms. So here they sort of show how this works. You have DNA cut at a certain sequence. It's called a restriction site. Or I'm sorry, you have restriction enzymes cut the DNA at a specific sequence called a restriction site that creates these sticky ends. If you cut DNA from a different source with the same restriction enzyme, then it's going to zip along the DNA and cut uh, along the same sequence. So then you have these complementary sticky ends. So then you can recombine DNA from two different sources, and then you can actually insert your gene of interest into the bacterial plasmid. Now, how do you get this stuff to join together? If you recall from the animation, there's another enzyme, I think it was like green in the animation. It's called ligase. So what ligase does is it forms a covalent bond between the sugar phosphate backbone, and then that sort of makes it one unit or one piece. Okay, so the big deal is you use the same restriction enzyme for both DNA sources. It cuts at the same restriction sites. You have complementary sticky ends. They can hydrogen bond with one another. Their bases can hydrogen bond, and then you use ligase to stitch together. When you do that, you create a recombined plasmid that has a gene of interest. So it's got some really cool capacities with this. All right, so when you create this plasmid, it's referred to as a vector. Think of a vector as being like a vehicle for some therapeutic gene or gene of interest. Okay, we can use viruses as vectors, like adenoviruses. Adenoviruses are being used to deliver some of the versions of the COVID vaccine, maybe the AstraZeneca virus or the AstraZeneca vaccine uses a virus as a vector because it can deliver the genes inside of a cell. We're in this section, we're going to be focusing on plasmids because, again, plasmids are what we're going to use in your bioluminescence lab or biofluorescence lab. Remember from the earlier chapters that bacterial cells can transform by taking up exogenous DNA. They can take up DNA from outside their bacterial cells. One thing they're really good at is taking up plasmids. So bacteria can take up plasmids from other bacteria, and in that way, it creates more variation within the bacterial population. All right, so again, a bacterial cell is going to have its one circular chromosome, so that's in sort of pink here. And then plasmids are little extra rings of DNA that can be shared. Okay, so a lot of times these plasmids will have genes on them that increase odds of survival for bacteria. A lot of times they'll have like genes for antibiotic resistance. And then what bacteria can do is copy those plasmids and then they can share them with their neighbors. So antibiotic resistance can actually spread through bacterial populations pretty readily by them sharing these plasmids with one another. They can copy the plasmid and then share it with a neighbor. The process is called conjugation. To conjoin means to join together, right? So what the bacterial cells can do is actually form like a little protein tube between neighboring cells called a conjugation tube or a conjugation bridge in this slide. But what happens is it forms basically like a little walkway between the bacterial cells. And so one cell can copy its plasmid and then share it with a neighbor. And in that way, again, it can confer like antibiotic resistance between neighboring cells. So this is called horizontal gene transfer or lateral gene transfer, where like the DNA isn't being sent from one generation to the next, it's sent from one peer to the next. So again, bacteria are really good at sharing plasmids. All right, so what are some of the key features of a plasmid? Well, one is you want to have an origin of replication. The origin of replication is where the DNA polymerase can attach and basically copy the plasmid. So you definitely need to have an origin of replication so you can copy the plasmid. Additionally, you're going to have restriction sites. So this is what's called a restriction map. So this is basically showing a plasmid, and it's telling you, okay, it's about 4,400 base pairs in length or in size. And then here are different restriction sites. So there are different restriction enzymes that will cut the plasmid in different locations. So what you can do is find some therapeutic gene or gene of interest, and you can cut that gene of interest on either side. And then if you have restriction sites that open up the plasmid, well, then you can again create those sticky ends that will join together, and you can stitch together your gene of interest into your plasmid. Another thing you want is what's called a screening gene. So in this case, it's an ampicillin resistance gene. So this is a plasmid that will allow the bacteria to grow in the presence of the antibiotic ampicillin. So what you'll do is you'll have the ampicillin resistance gene, and then maybe let's say you cut with EcoR1, you'll open up the plasmid. And then if you have your gene of interest and you cut that with EcoR1 as well, you can stitch together the gene of interest into your plasmid. And then that way, only the bacteria that have the plasmid can survive an ampicillin. So if the bacteria take up the plasmid, they'll survive in the presence of ampicillin, and they'll have your gene of interest because it's on the plasmid as well. And so the bacteria crank out what you're interested in. So that's what we're going to be doing in your biofluorescence lab is we're going to take a plasmid that has the gene for phosphorescence in it, and it's also going to have a gene for ampicillin resistance. So only the bacteria that take up the plasmid will survive on these little petri dishes that have antibiotic, that have ampicillin on it. So it's a way to screen out the cells that don't take up the plasmid. All right, so what's the benefit of being able to do this? Well, again, because of sort of the universal nature of the genetic code, you can cut human genes out, insert them into plasmids, and then you can have E. coli crank out human proteins. What has made Eli Lilly a lot of money over the decades is using this recombinant DNA technology to produce insulin. It used to be that insulin had to be collected from slaughtered pigs. You'd have to try and collect the insulin from like the pancreas of slaughtered pigs, which takes a lot of pigs and is really time consuming. It's expensive. But what Eli Lilly was able to do was put a human gene for insulin production into a plasmid and then have big vats of bacteria crank out the insulin, and they're able to make the insulin much more cheaply. So a lot of utility in being able to use this kind of recombinant DNA technology. All right, we'll pause here for this video, and then we'll jump into the other forms of technology shortly. Thank you.